Good afternoon.
Good afternoon,
Good
sir.
afternoon, sir.
Good afternoon, sir.
Well, in the previous classes, we have discussed many aspects regarding the microorganisms. And in that one, we have seen that number of organisms, they are pathogenic in nature. So they cause disease in the human beings as well as in the other animals. And some even cause the infection even in the plants as well as in the other microorganisms also. So when it comes to the pharmaceuticals, the pharmaceutical formulations, some of the pharmaceutical formulations are meant to be injected inside the body, or they have to be applied onto the wounded skin or the broken skin, or they need to be applied or inserted in the mucous membrane. In such case, if such products are being contaminated with any of the pathogenic microorganisms, so that will lead to the disease condition in the patient. So patient who is consuming the medicine for the sake of getting recovered or for the fighting back against these infections or anything, so in fact, so he will be getting more infected with some pathogenic or deadly microorganisms. So that is the reason. So these pharmaceuticals, some of the pharmaceuticals, so they've been described as the sterile pharmaceuticals or the sterile dosage forms. It could be injections, eye drops, or the certain ointments which just need to be applied on the skin or the broken skin or the wounds. So they are meant to be free from any microbial contamination. And that process is, we call it as a sterilization process. We apply number of different sterilization process based on the ability of the product to withstand the temperature or not, or some chemicals or not. They're based on that one. So they'll be subjected for the sterilization process. And this sterilization process is meant for these products so that there should not be any contaminating organisms. And once the sterilization is completed, once it is done, the next step and a very important step is the testing. Test for sterility. We call it as test for sterility. Because for any pharmaceuticals, so ultimately before the formulation reaches the patient, it will be undergoing a quality control test. And this sterility test is one of the quality control tests for the sterile dosage forms. So here we are going to check whether our product has been sterilized or not. I have repeatedly telling you the sterilization process is a all or none process. Either your product is sterilized or it is not sterilized. We cannot say what is the percentage of sterilization. So it is 80% sterilized, 90% sterilized, not even 99% sterilization. So that is not acceptable. So only thing is whether our product is sterilized or not sterilized. And if I tell you in the technical terms, whether the product meets with the sterility assurance level or the required SAL, whether it is meeting or not. So that has to be tested on the product. So after the sterilization. So how this sterility testing is done? The principle is very simple. Just take this sample, put it in an appropriate media, and incubate it at the appropriate temperature. And if in that media, if there is a growth of any microorganism after 24 hours or 40 hours or in any given stipulated time, so your product does not meet with the sterility test. That means it will fail the sterility test. And if there is no growth is observed in the product or no growth is observed in the media, so then your product will pass the sterility testing. This principle is very simple, but it is not as easy as it looks, because during the sterility testing, we do not test the whole sample for the sterility. For any quality assurance or quality control purpose, we do not take the all units for the sterility testing or all units for the testing. Suppose you have manufactured around 10,000 vials, it is not possible to test all the 10,000 vials. So, so what happens here is that we are going to take certain samples. So these, from where we take the samples. So how many samples has to be taken? And whatever the samples we take, and those samples we test for the sterility, and that samples, whatever the result comes from those samples, so that will be depicting the whole batch of that particular product. If those samples fail the test, the whole batch fails the test. If those samples pass the test, the whole batch will pass the test. So our results are based on the few samples which have been taken up from a lot for the testing purpose. So that is the one thing. So the difficulty is that we are not testing all the units. And the second thing is that there could be possibility of a false positive or a false negative report. What is the meaning of false positive report? Suppose you have taken the samples are being tested for the sterility. So when the sterility testing is being done, so if by chance during the testing, if some contamination occurs, so definitely the final result, it shows that there will be a growth of the microorganisms. And that growth is because of the contamination during the testing, not from the product. But what we get the result is that the okay, product is contaminated, so product doesn't pass the sterility testing. This is called as a false positive test. That means even though the product is sterilized, the report says it is not sterilized. Then what about false negative? Suppose when we put the sample in a media, of course, here one thing you should remember that whatever the product we are taking for the sterility testing, so in that one, even though if the microorganisms are present, if sterilization is not complete, so even though some organisms or viable organisms are remaining, so those organisms, the number will be very, very minimum, first thing. And second thing, they have encountered a very harsh environment, and so that they might have become very weak. So now during testing, if your media, the media in which we are going to place this sample to see whether the organisms are present or not, if they are present, whether they will grow or not. So in such case, if the media is not proper and if that your media doesn't support the growth of those weak and very few number of organisms, definitely you are going to get the negative test. That means sterility test passes. But in fact, there will be some organisms remaining, so which might have not grown during the testing period. So this is what we call as this false negative report. So these conditions, these problems, always they encounter and they will lead to the false reporting of the sterility testing. And the product, either it may be withdrawn or it may be taken for one more processing of sterilization, or it may be sent to the market without a proper results. So in order to avoid these conditions, there are certain guidelines given by the Pharmacopoeia Commission and other regulatory authorities to how to perform the sterility testing. Even though the basic principle, it looks very simple, but there are certain guidelines we need to understand and we need to follow these guidelines during the sterility testing. So today in this class, we are going to discuss regarding these this particular topic of the sterility testing of pharmaceuticals. So the content of this lecture is the introduction for the sterility testing, the definition of sterility testing, important guidelines, sample volume, sample size, and facilities and the media type required for the sterility testing, and two methods. One is membrane filtration method, another direct inoculation method. So how to perform these methods and what are their advantages and the limitations of these methods, it will be discussed. So sterility, it can be, we have already defined many times regarding sterility. So it can be defined as the freedom from presence of a viable microorganisms. If your product is said to be sterile, so then it should be free from any viable microorganisms. We don't say free from the microorganisms, we say free from the viable microorganisms. The reason is because only the viable microorganisms, they cause the problem. So during sterilization, they'll be killed. So it should be free from the viable microorganisms. But however, the conditions that guarantee the absolute sterility are usually too harsh for the active ingredients, and the definition of sterility for a medicinal product must be defined in the functional terms. That means sometimes it may be the temperature or it may be the radiation, whatever we use. So most of the time, so these conditions will be very harsh for the, if you want to kill all the organisms, it will be very harsh even on the product also, active ingredients. So that is the reason we usually talk in terms of the functional terms regarding the sterility. So in pharmaceutical practice, a container is defined as sterile when the probability is less than 1 out of 1 million that it is contaminated with the replicating microorganisms. Suppose you produce a 1 million units in a batch. In that one, there should be possibility of one less than 1 unit getting contaminated with the microorganisms. That is what we call it as a sterility assurance level. And of course, so whatever the care usually taken and the other precautions what we take, so even that will also be not possible. So even not even a single unit will be contaminated in that out of that 1 lakh. But for a practical term, we use this particular word. So that is called what we call as the sterility assurance level, the probability of less than 1 unit contaminated in 1 million. So because it is not possible to open and test each containers of the biological medicinal products, a number of samples, a representative of the lot being tested and taken at different times during the filling operations must be used to monitor the sterility. So as I have told you already, so we cannot test each and every unit of a lot or each and every unit of a batch. So only the representative samples which have been taken from that particular batch, they will be subjected for the sterility testing. A sterility test may be defined as a test that critically assesses whether a sterilized pharmaceutical product is free from contaminating microorganisms. So according to the Indian Pharmacopoeia 1996, the sterility testing is intended for detecting the presence of viable forms of the microorganisms in or on the pharmaceutical preparation. So the main purpose of the test is to determine whether the sterile product containing any viable forms of the microorganisms or not. So the sterility test, it's a quality control test used as part of product release for product required to be sterile. So before you release the product, which is said to be a sterile product, so it has to be subjected for this test, which is a part of quality control test, and this is called as the sterility testing. But this sterility testing has the significant statistical limitations. So it will really only detect the grass contamination. That means we cannot, as I told you once again, that each and every unit is not being tested. Suppose if I say that our product, we are having around 100 liters of a solution, liquid, which is been sterilized, and then it is to be filled in the containers, the final containers. So in this 100 liters, suppose if we expect around 100 microorganisms, a contaminating microorganisms number is 100. So this 100 liters is filled in a vials of maybe 1 ml or 2 ml or 5 ml vials, it will be filled. So how many vials you may be having? Maybe a 10,000 or 1 lakh vials will be present. Now, what is the expectation? So where does these 100 microorganisms will go? Whether all the 100 organisms will be present in a single vial, or 100 different vials will be containing each organism, or 50 vials will be containing two organisms each. So what do you expect here? Can anybody tell me? Can we say that where are those 100 microorganisms? Out of that, maybe let us say there are 10,000 vials which have been filled out of that final product which is sterilized, which is contaminated with 100 bacteria. So where those 100 bacteria will be present? Can anybody tell me? Is it possible to tell this?
No, sir.
It is definitely, it is not possible. So they may be, all the 100 may be present in a one vial, or they may be distributed in the, among the different vials. So that is the reason. So this sterility test, it does not give you the complete picture about it doesn't indicate regarding the complete sterility of the product. So it has got only the statistical, it has significant statistical limitations that it will give only the cross contamination. So even in a, if you are taken whatever the sample has been taken, in that one, if one or two samples, if they show the presence of these organisms, then the whole product will be considered as not sterile. But if you are missing that one, then definitely the product will be considered as sterile. So that is the reason there are lot many guidelines are issued. So how to perform the sterility test and on what basis the test, when you get the test is passed, so on what basis it has to be believed or it has to be reported. So now coming to these absolutely important guidelines. So why it is mentioned absolutely important guidelines is that it is a must. These guidelines must be followed. So there is no other alternative. You cannot make any changes in these guidelines. So the sterility testing, due to its inherent nature, is intimately associated with a statistical process wherein the portion of a batch is sampled almost randomly, right? So here we are not going to take all the samples, all the units are not taken. Only the random samples will be taken, and our report should be based on the certain statistical analysis. And hence the chance of particular batch duly passed for the actual usage. So only it depends upon the sample having passed the stringent sterility test. So your sample passes the test, the whole lot passes the test. Your sample fails the test, the whole lot will fail the test. So the sterility test should be performed under the conditions designed to avoid the accidental contamination of the product under investigation during the test. So this is the very, very first thing. First guidelines is that there should not be any accidental contamination of the product during the test. So the test has to be performed in such a way that, so there should not be any cross contamination during the testing. And nevertheless, the such particular precautions precisely taken for this purpose must not, in any case, adversely affect any microbes that should be revealed in the test ultimately. The one thing is, the very first care what we have to take is that there should not be any cross contamination during the investigation or during the testing of the product. Second thing, so you have taken at most care, the maximum care has been taken, sterilization and other things, aseptic condition, everything. So these conditions are what we are expecting, or these precautions what we are taking. In any way, it should not adversely affect the growth of the organisms if any are present in the product. Is it clear? Is this clear? Say yes or no, so that I can continue. Unless you understand this particular part, it is not possible to understand the sterility testing.
Sir, can you explain the second point?
Yes. See, what is the first point? That we have to take utmost care so that our product should not get contaminated during the testing. So we have taken the sample and samples are already sealed and everything is fine. Now, while testing, there should not be any cross contamination during the testing so that you will get the false positive report. At the same time, the test should be conducted in such a manner that if there are any organisms are remaining in the product unsterilized, okay, remember, if any product, your product is unsterilized, means it is not been properly sterilized and some viable organisms are remaining in that one, and those organisms should not be adversely affected during the testing. That means we should not inhibit their growth during the testing, so that otherwise what happens, you will get a false negative report. That means your product will, the report says that the product passes the test, the product passes the sterility test, but in fact there will be some viable microorganism present in the product. So these both conditions, the both cases should not happen. Now is it clear?
Yes, sir.
Yes, sir.
Okay. So working environment wherein the sterility tests are meticulously carried out must be adequately monitored at the regular intervals by sampling the air and surface of the working area by performing the necessary control test. So this is for maintaining an aseptic condition while testing the product in the area. So the working area should be adequately monitored at the regular intervals. Regular intervals means usually before testing any sample, so the area should be checked for presence of any microorganism. That means it should be sanitized, it should be sterilized, the air should be sterilized. We have to work in a laminar flow hood and everything, all the care has to be taken. And sterility tests are exclusively based upon the principle that in case the bacteria is strategically placed in a specific medium that caters the requisite nutritive material and water and maintained duly at a favorable temperature, usually it is 37 plus or minus 2 degrees centigrade, the microbes have the tendency to grow and their legitimate presence may be clearly indicated by the presence of turbidity in the original clear medium. So simple, very simple principle, what I told you in the earlier case. If your product is being contaminated with any microorganism or the live microorganisms, then if you put that product in a media, a specific media, at a favorable temperature, if you incubate it, so definitely these organisms will grow, and that growth will be evident by the presence of the turbidity. You can see here. So this is the initial test tube, which is a very clear media. And then if there is a growth, then the whole media, it will be turn turbid. So now if it turns turbid, that indicates that the product contains the viable organisms. And if there is no turbidity, the product does not contain any viable organisms. So this is the result, final result. But because we should not get the false positive and false negative report, we should take utmost care. So the extent of probability in the detection of the viable microorganisms for the test for sterility, usually it increases with the actual number of supposedly present in a given quantity of the preparation under the examination, and is found to vary according to the species of the organisms present. So what is the probability of detecting these organisms if there are any microorganisms are present? One thing is the actual number of may be present, whatever the number may be present. So I have told you, if in the whole lot, if it is containing the 100 microorganisms, where are those 100 microorganisms? Whether they are present in a single vial, whether we have taken that particular vial or not during the sampling, if they have been distributed among other 100 vials, suppose the maximum probability, and is there any sample has taken from those 100 vials? So it all depends on those numbers and also based on the species of the organisms which is present. So however, extremely low level of contamination cannot be detected. As I told you, it's very difficult. So in 100 liters, suppose if we expect that there may be some bacteria still remaining, some 10 viable bacteria still remaining in the sample, in the product in 100 liters. What is the probability of finding those 100 microorganisms? Is very less. It's very, very less. So that 100, even if you think that those 10 microorganisms, they are distributed in 10 vials, you are having 10,000 vials. Out of that 10,000 vials, the probability of taking those 10 vials, one among those 10 vials as a sample, what is the probability? So we don't know. So it is very, very remote. So that is the reason. So extremely low level of contamination cannot be detected conveniently on the basis of random sampling of the batch. So sampling is very, very important here. So how this sampling will represent the whole lot, we'll come to that later. So in case the observed contamination is not quite uniform throughout the batch and random sampling cannot detect the contamination with absolute certainty. So if the contamination is not uniform, if those 10 bacteria, they're remaining in one place or they're being distributed, so it all depends. So observed contamination is not quite uniform throughout the batch. The random sampling cannot detect the contamination with absolute certainty. And therefore, the compliance with the test for sterility individually cannot certify the absolute assurance of freedom from the microbial contamination. So what is the meaning of this? Only sterility testing does not give you the guarantee of your product to be sterilized. You manufacture the product, then you subject it for sterilization, and then you test it for the sterility. Okay, the result comes, sterility test passes. But what is the guarantee? Because I have already told you what are the possibilities, what could go wrong during the sterility testing. So what is the guarantee that when your sterility test passes also, what is the guarantee that the whole batch is sterile? So that comes here. So nevertheless, the greater assurance of sterility should invariably originate from the reliable, stringent manufacturing procedures, vice versa, strict compliance with the good manufacturing practices. So for manufacturing and sterile product, we are having a particular GMP. We are having a particular SOP. So there are certain procedures, set procedures for manufacturing the sterile products. There are set procedures for sterilization of these products. So when you manufacture it with a proper GMP, the chances of contamination in that product will be very, very less. Second thing, when you are subjecting it for a strict sterilization process or a very effective sterilization process, so definitely if there are any, whatever the number of contaminating organisms are there, definitely they will be killed and it will be free from the contamination. And third thing, the validation. So what we have seen earlier. So on what basis we believe it? Because we have validated the sterilization process. Whatever the sterilization process we are conducting, so that process has been validated and it has been recorded. Based on the validation report, based on the GMP report, and based on whatever you have followed during the manufacturing, and finally based on the sterility testing. So by comparing all these parameters of all these documents, then only we can say, yes, the product has been sterilized based on the sterility testing reports. So only sterility testing report does not give you any guarantee regarding the sterility of the product. It all based on the GMP, the sterilization process, and the validation of the sterilization process. So tests for sterility are adequately designed to reveal the presence of microorganisms in a sample used in the test. So however, the interpretation of the result is solely it's based upon the assumption that the content of each and every container in the batch, had they been tested actually, would have complied with the test. So that means it should, whatever the sample you have taken, it should represent the whole batch. So as it is not practically possible to test every container, a sufficient number of containers must be examined to give a suitable degree of confidence in the ultimate results obtained for the test. So depending on the batch size, if you are having, what is the batch size? If the batch size is a 10,000 or it's a 50,000 or a 1 lakh. So based on that, on the sufficient number of samples has to be taken so that it should represent the vast range of the lot, a vast range of the batch. So it has been duly observed that there exists no definite sampling plan for applying the test to a specified portion of a discrete units selected carefully from a batch is capable of demonstrating that almost all the untested units are in fact sterile absolutely. So there is no definite sampling plan. So there may be different pharmacopoeias, they give the different plan for this sampling, but there is no definite sampling plan because none of them, they give the absolute surety that your samples, whatever the samples you have collected, it will represent the whole batch. So therefore, it is indeed quite pertinent that while determining the number of units to be tested, the manufacturer must have the adequate regard to the environmental parameters of manufacture, the volume of preparation per container together with the special considerations specific to the preparation under investigation. So once again, so how the product has been manufactured, how it has been sterilized, everything has to be taken into the consideration before giving the results. So then the first thing comes the sampling. So sampling is now you might have understood what is the importance of the sampling, because the samples what we are going to take are the few numbers compared to the whole lot, and those samples should represent the whole lot here. So sampling, the number of containers and volume to be tested defined in the pharmacopoeia. So the different pharmacopoeias, they give the different guidelines for how the sample has to be taken. Samples from aseptically manufactured product should be taken from the beginning, middle, and end of the batch fill, and also after the interventions and the stoppages. That means when sterilize a bulk of the product, bulk product is sterilized, and now it has to be taken for the filling. So bulk, your product is sterilized, and the filling materials are sterilized. Now the filling has to be taken place. So during the filling, utmost care has to be taken now, because it is not terminal sterilization, but it is the sterile product is being filled in a sterile containers. So maximum care has to be taken with zero margin of error. So when you take the samples, you have to take in the beginning of the filling session, in the middle, and at the end of the batch fill. So 3 times. And in between, whenever there is an intervention, if because of any breakdown or anything, if the filling stops or for anything, any filling stops. So in between that interventions and the stoppages also, during that time also, the sample has to be taken, so that there should not be any contamination during that time. And samples from terminally sterilized product should be taken from previously identified cool spots within the load. So now, if you are taking the samples from a terminally sterilized product, that means you already filled everything and then you are going to keep it for the sterilization. When you are taking it for the sterilization. Now, the samples have been completely filled or they are sealed and then they are sterilized. Now, from where we have to take the sample? So here we have to take the samples. So where those samples were, the containers are kept in a cool spots within the load. In the validation of the sterilization process, I have told you regarding identification of the cool spots in an autoclave. So from that place, whichever the samples are placed in that particular, located in that place, so we have to take the samples from that particular spot. That is called as the cool spots. These cool spots are nothing but they'll not be any, it doesn't mean that it will be it does not have any temperature, but it will receive the temperature slowly. So that's why we call it as a cool spots. So from that spot, whichever these units were kept in that spot, they have to be taken for the sampling. And sampling should be sufficient to allow for the retest if needed. So now whenever we do the sampling, instead of taking 1, we have to take 3 or 4, so that if any retesting is required once or twice, so the sample should be sufficient. So according to the pharmacopoeias, so how the sample volume in sterility testing is taken. So as per the USP, United States Pharmacopoeia, this is the sample volume for sterility testing. So how much of sample has to be taken for the sterility testing? So if the container content is 10 ml or less, so that means the content of the unit is less than 10 ml or less than 10 ml, then 1 ml of the total content if less than 1 ml. If it is less than 1 ml. So if it is less than 10 ml, then you have to take the 1 ml of the sample, 1 ml of the sample taken for this testing. And if it is less than 1 ml, the whole content, the total content has to be taken. And the minimum volume of the media used should be 15 ml here. And if it is between 10 to 50 ml, the content is 10 to 50 ml, then we have to take the 5 ml of the sample is taken for the testing and then 40 ml of the media. And if it is more than 50 ml, the container content is more than 50 ml, then we have to take 10 ml of the sample and it is to be put in the 80 ml of the medium. And in case of the solids, if it is less than 1 ml or 50 mg, so in such case, the whole content for each, this is as per USP, and now as per European Pharmacopoeia, less than 1 ml or 50 mg, then the whole content for each batch and for the fungal test, and the amount of media should be not less than 10 times of the volume of the sample. So suppose if you are taking 1 ml, the volume of the media should be 10 ml. If you are taking 2 ml, the volume should be 20 ml. That means not less than 10 times of the volume of the sample. And if the container content is less than, the 1 ml or 50 mg, but less than 4 ml or 200 mg, the half the content for each test will be taken. And similarly, for between the 4 ml to 20 ml and for more than 20 ml, so these pharmacopoeias, they form a different pharmacopoeias, they will specify how much of the volume to be taken for the testing. And then coming to the sample size. So how many samples has to be collected from the bulk? So if the containers in a batch, if it is not more than 100, if it is less than 100 or less than 100, then 10% or 4, whichever is more, we have to take. So that means if there are 100, if there are 100 containers are there in a batch, then we have to take 10%, that should be 10. So suppose if the container numbers are in a batch, if it is less than 40, suppose if we say 30, so in that case the 10% will be 3, so we are not taking 3, instead of that we are taking the 4. So like that, if it is more than 100 but not more than 500, we have to take the 10 samples, and more than 500, 2% of the containers, number of containers are the 20, whichever is less, so that has to be taken. That is as per the European Pharmacopoeia. And as per the United States Pharmacopoeia, so once again, this United States Pharmacopoeia, the number of samples to be taken is based on the testing, based on the sterilization method which has been adopted. Suppose if you have adopted the sterilization method of autoclaving at 121 degrees centigrade for 15 minutes, the number of samples will be 10 will be sufficient. Whatever may be the container size, whatever may be the batch size, the 10 samples will be sufficient. And if they are sterilized in the final container, but not autoclaved at 121 degrees centigrade for 15 minutes. If you have used any alternative method of sterilization or alternative temperature time cycle, in such case, the number of samples should be more. So we are taking 20. If you have used the biological indicator during the sterilization process, then the 10 samples will be sufficient, and not sterilized in the final container. If the bulk is sterilized and then filled in a sterile containers, then the sample size will be definitely more. It will be 30. And if the bulk has already passed the test for sterility, then 20 samples will be sufficient. So this is depending on how we are going to do the sterilization. So as per the United States Pharmacopoeia. So like that, the different pharmacopoeia, they give the different guidelines for how much of sample has to be taken, sample size should be collected, and then how much of sample to be used for the sterility testing. And this is as per the Indian Pharmacopoeia. For injectable preparations, so once again, the same thing, not more than 100 containers, either 10% or 4 containers, whichever is greater, we have to take. So almost similar to the USP. And here the pharmacopoeia specifies the batch may be defined for the purpose of these tests, a homogeneous collection of these sealed containers prepared in such a manner that the risk of contamination is the same for each of the units present in it. That means if there any risk of contamination is there, the whole batch should have the same risk of contamination. So then only it is called as a batch, a single batch. If some of the part is having different risk and another part is having different risk, so in such case, they will be, it is not considered as a single batch. So when you consider as a single batch, it should be a homogeneous collection of the sealed containers, so which are having the same risk of contamination. And for the ophthalmic and other non-injectable preparations, surgical dressings and bulk solids, so the different parameters or different methods of the different sampling will be, it is given as per the Indian Pharmacopoeia. So this you have to go through it. So you may be asked in the examination to write down the what is the sample size as per the Indian Pharmacopoeia. How much of sample is to be taken as per the Indian Pharmacopoeia? You have to mention for the different IP, these injectable preparations, that is injectable preparations for ophthalmic preparations, and for surgical dressings, and for the bulk solids differently. Now coming to the facilities. So facility is a very, very important aspect, because we need to conduct this sterility testing in utmost care. Sterility testing should be carried out under the same condition as the aseptic manufacturing. So whatever the conditions have been used for the aseptic manufacturing, that means with the zero tolerance level for the microbial contamination. So in the same condition, the testing also has to be done so that there will not be any cross contamination during the testing. So we have to use the grade A laminar airflow cabinet and grade B background. So you'll understand this in the later stage. So when you study regarding the laminar flow chamber, grade A laminar flow chamber means which will be fitted with the HEPA filter and UV light and other thing, and may be carried out in an isolator. So the maximum care has to be taken during the testing. And air supplied always through the HEPA filters, high efficiency particulate air filter. So with the air which will be free from the any microorganisms. And for that HEPA filters, the pressure should be monitored and it is alarmed. So even the HEPA filter is not only HEPA filter is not only important. So the pressure of the air which is coming from that HEPA filter is also very important. So if the pressure drops, then chances of contamination will be more. So that's why there will be a pressure alarm will be present in the laminar flow chamber. If the pressure goes down for the air which is coming from this HEPA filter, if the pressure goes down, then there will be alarm which will blink or it will give the sound and it will give the indication regarding the drop in the pressure. The access area should be through the airlocks. So what we call it as a double door entry. So this through the airlock means so outside the pressure will be less and inside the air pressure will be more. So when you open the door, the air from inside to outside it will go, but not from outside to inside. So that is the airlock system, and also there will be a double door entry. So that's what is called as aseptic manufacturing condition. So in that one it should be done. And operators should be appropriately gowned in sterile garments to avoid any cross contamination. And operators should be appropriately trained and validated. So the training of the personnel who is conducting this test is very, very important because one should know the consequence of the results. If any false results are given, so what is the consequences? They should know and they should be properly trained. And appropriate cleaning, sanitation, and disinfection procedures should be in place and it should be documented. And environmental conditions should be conducted. Monitoring should be conducted. That means the presence of the air samples has to be taken, the surface samples has to be taken for any possible presence of the microorganisms in that area. And next one is the media type. So this is another important factor because as I told you earlier, if there are any microorganisms are remaining in the product, unsterilized means in an unsterilized product. So these organisms will be very, very less in number and they will be under a shock because they have gone through a very serious, severe process of sterilization. So these organisms, even though they are alive, so their recovery rate will be very, very less. So that's why the media what we use, it should be sufficient or it should be in such a way that it should be able to help these organisms to grow back. So what we use is usually soybean casein digest media, also known as the trypticase soya broth. And we use the fluid thioglycolate media. It is usually used for detecting the aerobic and anaerobic organisms. And validation studies should be demonstrated that the media are capable of supporting the growth of range of low number of organisms in the presence of the product and may need to incorporate the inactivators. So here we should know that we have to validate even these media also. So whether these media are having the ability to support the growth of that low number of the different range of the microorganisms. They cannot be a selective media. They cannot be a selective media. So they should allow the growth of any form of microorganisms, especially the bacteria, both the aerobic and anaerobic bacteria. So they cannot be selective in nature. And also they should be able to facilitate the growth of small number of these organisms. And the growth should be evident after 3 days, usually in case of bacteria, and 5 days in case of the molds. And media may be purchased or made in-house using the validated sterilization procedure. So you can prepare the media in-house in the industry itself, or it can be purchased from the vendors with the certificate. And should be tested for growth promoting qualities prior to use. So definitely before testing the media, first of all, the media should be tested whether it is having this growth promoting quality or not to allow the growth of this low number of organisms, and should have the batch number and shelf life assigned. So definitely that should be present. So this is regarding the sterility testing. So what are the requirements? So what are the precautions has to be taken, and what sort of media should be used. So this is the thing. And then coming to the methods as defined in the pharmacopoeia sterility testing, there are two methods. One is called as the membrane filtration technique. Another one is the direct inoculation, or is an alternative technique. So based on depending on the nature of the product, so either one of these method will be used. Suppose we are having an injectables, an aqueous solution, or it may be a non-aqueous solution. Injectables, it's a liquid, so then definitely we go with the membrane filtration technique. But whenever it is not possible to go with the membrane filtration technique, suppose we are having an the injectable, that is a syringe. Syringe is a sterile product, isn't it? Dispovan, you might have seen. So, is it possible to filter the dispovan for the sterility testing? Dispovan syringe, can you test it by filtration method? Can you test the syringe by a filtration technique?
No.
So definitely no. So then in that case, we have to go with the alternative method. That's what we call it as the direct inoculation method. So these are the two different methods which will be followed for the sterility testing. One is membrane filtration technique, another one is the direct inoculation technique. So that which technique has to be used, so that depends on the nature of the product. So this we are going to study in the next class, probably on the Monday. So Monday it will complete this sterility testing chapter. So this is it regarding till the for the today's class.
So for test.
Case, so we'll stop the class for today. Best of luck. Study properly for the test for the Tuesday. Take care. Bye.
Okay, thank you, sir.